Today we will talk about the neurotransmitter gamma-aminobutyric acid, or GABA. To begin, we will talk about its structure. GABA is a modified form of butanoic acid, which is a short-chain hydrocarbon made of butane, in which one of butane's methyls, or CH3s, has been oxidized into a carboxyl group. A carboxyl group is the functional group responsible for making this an acid. GABA also has an amino substituent located at its fourth carbon. GABA is made by a chemical process derived from cellular respiration. Specifically, it starts with alpha-ketoglutarate of the Krebs cycle, which is also known as the citric acid cycle. Alpha-ketoglutarate is modified by the enzyme alpha-oxaloglutarate to create glutamic acid. The decarboxylation of glutamic acid is synthesized by another enzyme called glutamic acid decarboxylase. The product is the GABA molecule, which among other things is a neurotransmitter. This process is called the GABA stunt. It is a closed loop that preserves and produces GABA. GABA is present in high concentrations in many brain regions. It is about 1,000 times more concentrated than the classical monoamine neurotransmitter in the same region. This is because of the powerful and specific actions of GABAergic neurons. In these regions, GABAergic neurons are the neurons in which GABA neurotransmitters are synthesized and preserved. There are two types of GABA receptors in the brain, GABA A and GABA B. Let's begin with GABA A receptors. GABA A receptors are of the cis-loop ligand-gated ion channel superfamily. Cis-loop is referring to the 13 amino acid loop in its structure. The loop sits between two cysteine residues. The cysteine residues formed by sulfide bridges between one another. They are also ligand-gated ion channels, which means once activated, the GABA A receptors open some sort of ion channel on the surface of the neuron. In this case, it is a chloride ion channel. GABA A also has a subclass called the GABA AP receptor. The only difference is a P subunit. This subunit makes the GABA AP receptor impervious to many allosteric modulators that affect the GABA A receptor. Allosteric modulators are inhibitors that bind to the allosteric site of a channel to indirectly inhibit it through conformational changes. GABA B, on the other hand, is G protein coupled. As the name suggests, the G protein coupled receptors interact with a nearby G protein when activated by a transmitter. This activation causes a conformation in the receptor. G proteins are proteins in the plasma membrane with the ability to bind GTP, guanosine triphosphate, and GDP, guanosine diphosphate, both of which are molecules that usually act as signaling molecules in the
functions through two mechanisms. It either hyperpolarizes the postsynaptic membrane through an influx of chlorine ions or an efflux of potassium ions, or it decreases neurotransmitter release as GABA binds to G protein-coupled receptors in the presynaptic terminal, which cause a decrease in calcium influx. These mechanisms are what allows GABA to perform its functions, such as motor control, vision, regulation of anxiety, as GABA is induced by prolonged periods of stress, and other cortical functions. When talking about GABA deficiency, anxiety is the most common disorder associated with it, as GABA is believed to be needed to organize thoughts and emotions, and anxiety is generally an overstimulation of certain parts of the brain, which cause many negative thoughts and worrying. People with anxiety could be medicated through supplementation of GABA pills, or other compounds or minerals which help induce the release of GABA, such as magnesium. Some types of epilepsy are also linked with GABA deficiency, as similarly to anxiety, epilepsy is caused by overstimulation of regions of the brain. A special thing about GABA receptors is that they have various binding sites, and which makes the agonists for GABA not necessarily similar in structure, as GABA receptors have binding sites for ethanol and benzodiazepines. This helps explain how alcohol could induce a calming effect, as when the ethanol which is found in alcohol is ingested and reaches the brain, the ethanol will bind to these binding sites found on GABA receptors, thus inducing a similar effect to GABA release, which is a calming effect and a state of tranquility. It could even induce sleep at higher levels of when levels of ethanol are high. On the other hand, benzodiazepines are also have a specific binding site found on GABA receptors, but they act in a different way to alcohol and GABA, because when they bind to the receptors, the binding site on the receptor, it reinforces the binding of GABA and increases the likelihood that the effect of GABA will occur, thus causing more reliable effects. They could also be used as medications for diseases which are or disorders similar to anxiety. Thank you.